Tailoring Camelina sativa seed fatty acid composition via Fast Neutron mutagenesis. So he's treating these seeds and looking at the fatty acid composition thereof. So this could be a very powerful and interesting tool. So I'm looking forward to hearing this talk. Winston, please begin.
Hello everyone, I'm Xinjie. In here, I'm gonna do introductions of my poster. The topic of my poster is Tailoring Camelina sativa fatty acid compositions via Fast Neutron mutagenesis. And let's begin with the introduction. For camelina oil, the most amazing part is it contains a very high amount of polyunsaturated fatty acids, such as essential fatty acids like linoleic acid and alpha-linoleic acid, which contain about 20% and 33% of the total oil content, respectively. And Camelina sativa has a hexaploid genome structures. It means Camelina has 3 subgenomes, and this genetic redundancy will reduce the likelihood of lethal mutations. The methods we use in this project to introduce mutations is Fast Neutron remediation. And the major mutations caused by this type of mutagenesis approach is deletions. And this kind of mutations have chance to completely knock out genes in Camelina genome. Experiments in this project can be generally divided into 2 parts. The first part is phenotyping, and the second part is genotyping. For phenotyping, the 2 major experiments is germination test and gas chromatography analysis. Here shows the result of germination test. The first group is the control group, and other groups are the treated group. And we can see at 24 hours, higher dosage treatment has less seed germinated. But after 48 hours, all the group has a germination rate close to 100%. And this indicates that Fast Neutron treatment can slower the germinating speed of the seeds, but fail to lower the germination rate of the seeds. And this chart displays the comparisons of the fatty acids composition of seeds between mutant lines and reference. And here the reference is Midas. And we can see the 2 mutant lines I selected shown in this chart had a very different content of linoleic acid and alpha-linoleic acid compared to the Midas. And this kind of change of fatty acid compositions is desired, and the mutant lines contain this desired lipid profile will be used for further genotyping analysis. So the second part of the experiment is genotyping. The 2 major techniques we use is PCR scanning and genotyping by sequencing. For PCR scanning, it's used to detect whether there is deletions occurred into the Camelina genome. And genotyping by sequencing, also known as GBS, is more focusing on understanding what exactly happens inside Camelina's genome. And after this experiments, we also will do backcrossings. This backcrossing is in order to backcross the mutant lines with desired lipid profile back to the commercial Camelina species in order to induce those desired traits in the commercial Camelina species. and this can help to improve the camelina oil quality. And this will be all the content of my introduction. Thanks for listening.
Thank you, Winston. Very interesting talk, and
Thank you.
very a vast field of potential applications and research. So thank you very much. Sarah, did you have a question?
I did. First of all, I just want to say congratulations, Winston. I thought you did an amazing job presenting your research. I was wondering if you could explain more on your results slide where you compared the Midas control with your two tests. If you could explain more why the test in blue showed higher linoleic content and the test in orange showed higher linolenic content, if there was a reason behind that.
Yes, because for the mutant we treated with fast neutrons irradiations, and after treated with the mutation, the fast neutron irradiations, there could be some mutations introduced into the plant genomes, and if the mutation is occurred in the genes that associated with some kind of enzyme genes, like some genes associated with like fatty acids elongase or fatty acids desaturase, then this mutation may be block these genes expressions or if improve those genes expressions, then can lead to this different lipid profile when we do the GC experiment.
Thank you very much. Here's a question. Gondoic acid is a very uncommon fatty acid, and I wondered if you thought about prioritizing it because it has potential for use in cosmetics and as a transfer agent to bring things through the skin. Is that target been, have you been made aware of that potential target for a market for the altered camelina?
Thanks for your question first. This is a very good question. Yeah, and for gondoic acid, because this kind of fatty acid is not very reproducible, this gondoic acid is good for our human healthy. And so for industry, if we the main interest in the market is to have a much more healthier oil products. So we want to improve camelina oil qualities. We hope to increase like omega-3 and omega-6, this kind of fatty acids content into the oil products instead of others fatty acids. So for gondoic acid, because they didn't have some reproducible this kind of fatty acids have a healthy effect on our human body. So we hope to eliminate or reduce those accounts in the camelina oil products.
Okay, thank you very much. Susanna, did you have a question too?
Yes, thank you. Winston, congratulations for your very nice work. And I just would like to ask you, in all these mutants you got till now, did you have already one that maximizes in the yield of oil and fatty acid composition? That means it is the best in terms of oil yield and fatty acid composition, or is it not possible?
Yes, thanks for the question. For the we cannot say we can find the best or the maximum oil product, oil content of the seed, because we don't know what the mutation caused on this plant genomes. So it may show different phenotypes or genotypes after treated with fast neutrons. But so far we have many different phenotypes of these fatty acid compositions, like I showed in the presentation. Some have higher omega-3 or some have lower omega-6, something like that. And there could be some phenotypes like higher omega-9 or oleic acid, or even some seed treated with Fast Neutron, they can lead to low germination rate or other kind of phenotypes, like different visual phenotype, like the leaf will be a different shape. And also for the oil content, so far we don't find a light have a very high oil content, but the average oil content so far is about like 35% or something around this. And we don't know if we can find some amazing oil content mutanites later or in the future. So it's very hard to say which one is the best, or because they all have different characteristics.
Thank you. Congratulations.
Thanks.
Scott, may I ask a question?
Please, go ahead, Professor.
Okay, sure. Thank you, Winston, for presenting, and I also congratulate you.
Okay.
I guess, just if you can speak to why you chose fast neutrons to cause mutagenesis versus other methods. I wasn't sure if there was a specific hypothesis that you might be testing in your research work.
Yeah, we choose for this question, because we choose fast neutron radiation mutagenesis method to do this project is because fast neutron the major mutant type caused by the fast neutron is deletions, and for Camelina's genome structure is hexaploid genome structures, and so which means it has many genetic redundancies in their genomes. So if we can lead to deletions into the genome structure, especially some large deletions, it may have a higher chance to completely block or delete genes, some kind of genes. And compared to other methods like using the chemical mutagens like EMS, those chemical mutagenesis methods, the major mutation types caused by those methods is the single nucleotide substitutions. This kind of mutations is just change one single nucleotide, and this is very hard to determine, and also it may not have a very numerous influence on the phenotypes. And so we choose a fast neutron to have a try.
Thank you very much for your answer. May I ask one more question, Mr. Moderator, Dr. Moderator?
Please.
I was curious where you did this work at. Did you use the neutron facilities at Ottawa, or where did you do this work at? What was your neutron source?
We treated seeds in another institutions in the McAllen Nuclear Radiation Center at UC Davis in California, not in our center. Yeah.
Thank you.
Thanks.
All right, I think those are all the questions. Thank you very much, Winston, for a great presentation, and I thank you, all of our students. And now we're switching to the voting time.